So already we discussed about the basics of pharmacokinetics. At least I hope that you know the definition for pharmacokinetics, right? So if you take pharmacology, there are two major studies in pharmacology. One is called as pharmacokinetics, another one is the pharmacodynamics. So once if we intake any drug molecule, they have to undergo the pharmacokinetics and pharmacodynamics. The simple way to define pharmacokinetics is what the body does for the drug. Again, I repeat, the very simple layman definition for pharmacokinetics is what the body does for the drug molecule. There are four important parameters which are studied under pharmacokinetics: absorption, distribution, metabolism, and excretion. These are four major parameters which are studied under pharmacokinetics. So, what is pharmacodynamics? So, pharmacodynamics is what the drug does for the body. Again, I repeat, pharmacodynamics is what the drug does for the body. So, in one of the lecture, we discussed in detail about the drug action of aspirin, the molecular mechanism of action of aspirin. So, if you take aspirin molecule, drug molecule, so which is capable of reducing the pain, fever, inflammation. There are many properties which are associated, right? So, how the drug molecule produces those effect, biological responses. So, if you study about that, that comes under pharmacodynamics, right? So, the second unit is completely for pharmacokinetics, and the third unit is fully for the pharmacodynamics, right? So, in this unit, we'll be studying about, in detail about the absorption, distribution, metabolism, and excretion. What's the use of those parameter in terms of medicines? That is what you'll be studying in this unit, right? So let me start with very fundamental thing, right? So what is meant by permeation, right? So permeation, the dictionary meaning of permeation is the movement, penetration of the molecule, and this permeation of the drug molecule depends on various parameter. So here I have listed three important parameters which are related to permeation, which influences the permeation. Again, I repeat, permeation is nothing but the entry of the molecule. Okay, so it depends on some factors. The three important factors which are associated with permeation are solubility, concentration gradient, and surface area and vascularity. There may be other parameters, but these are the factors which are very important, very closely related to the permeation. So these are the factors which decide the permeation, rate of permeation, ability of permeation, whatever it is. These are the parameters which are very important. So the first one is solubility. Uh, so many times, I repeated this. If you say any molecule, drug molecule as a good drug molecule, then so those drug molecule should have both lipid solubility as well as water solubility. Again, I repeat, very fundamental concept. That's why we are repeating all these points again and again. If we say any molecule as a good drug molecule, in the first unit we studied about the importance of solubility, which functional group enhances the water solubility and which functional group enhances the lipid solubility, right? So we know that any good drug molecule should get soluble in both lipid as well as in water. One important point is that so the drug molecules will be having, we expect the drug molecule to have more lipid solubility than water solubility. Again, I repeat, don't get confused. The molecule should get soluble in both water as well as lipid. Which solubility we expect more? We expect the molecule to have more lipid solubility than water solubility. But anyway, the drug molecule should soluble in both lipid as well as water, right? So if the molecule is having lipid solubility, then those molecules will have ability to diffuse through the lipid bilayer. Again, same point many times, several times I have discussed. So one of the important barrier for the drug molecule is the cell membrane. So our cells are made up of cell membrane, that's outer coverage. So that outer membrane which separate the inside of the cell and outside of the cell. So the various contents which are present within the cell or preserved within the cell with help of this lipid bilayer. So which means that once if you break this lipid bilayer, okay, the cell will it's called as lysis, right? The content of the cell will come out and the cell will die, right? So if the molecule is lipid soluble, then they will be having ability to diffuse through the bilayer. So which means that they can enter into the cell. So that is why the lipid solubility is very important. Okay. So if any molecule which lacks this lipid solubility, then they may struggle to get enter into the cell, or they may not even able to enter into the cell at all, right? So, but for permeation, right? So the water solubility is also important. So once if you take any oral drug molecule, first the molecule should get dissolved in water. If you take the content of stomach, mostly it is water in nature, right? So the molecule should get dissolved in water. So then only the permeation, the absorption process will takes place, right? So the permeation that depends on solubility. So solubility in both lipid and water is expected. The very first solubility what we expect is on the water. So then only it can enter into the cell, for which the lipid solubility is again important. The second important parameter is the concentration gradient. So concentration gradient is another factor that determine the permeation. So what is this concentration gradient? So this is the movement from higher concentration to the lower concentration. That is what exactly here the meaning of concentration gradient. So normally, if you take some room spray as an example, right? So room freshener, if you spray the room freshener in one corner, right? So after some time, that will spread throughout the room. If you spray only in one corner, right? So it will spread throughout the room. The entire room will be spreaded with the molecules which are present in the room freshener, right? So how that is happening? That is happening through a process called as diffusion. The molecule diffuse. So in which direction the molecule diffuse? So it is diffusing from the higher concentration to the lower concentration. All these are not new for you, right? So you might already studied all this in your 9th standard, 10th standard, or 11th standard textbook, right? So that is what is the meaning of concentration gradient. So always you have to keep in mind that the movement will be from the higher concentration to the lower concentration. Okay, that is what I put up here as the diffusion down a concentration gradient, movement from higher concentration to the lower concentration. So for this type of movement, there is no need to supply any energy. It is a spontaneous reaction. It takes place on its own without taking any energy. So the point to remember is that only free non-ionized form of drug molecule can contribute for this concentration gradient. So now you have to relate some concept what we have studied in the first unit. So we made some calculation related to how much amount of the drug molecule will be present in its ionized form, how much amount will be present in its non-ionized or unionized form, with the help of Henderson Hasselbalch equation. So the relationship between pKa, pH and pKa, right? So here the concentration gradient, okay, so which type of drug can contribute? Only the free and non-ionized form of the drug molecule. So we already studied that the ionized form of the molecule will not contribute for entering into the cell. It can contribute for elimination of the drug molecule. Just like that, one more point that we not yet studied is that only the free drug molecule. So you may have a doubt that what is this free drug molecule? So once if you take the drug molecule, right? So once if it enter into the circulation, blood, then the blood will have several protein molecules. Again, I repeat, not inside the cell. The blood itself will be having several protein molecule. So this drug molecule can go and bind to those protein. So there are two chances for the drug molecule. One is the bound form, another one is the free. The free is the drug alone, not bounded to any protein molecule. The bound form is the one which is bounded to the protein molecule. So for the permeation and for the concentration gradient, only the free drug molecule they can contribute for the concentration gradient, not the bound one. Is that understandable? The third important parameter that influences the permeation, the factor that influences the permeation is the surface area and vascularity. Very important parameter. So if you think of our body, right, certain regions of our body will be having larger surface area and greater vascularity. Certain regions may have a smaller surface area, and certain tissues they may have lesser vascularity. So surface area and vascularity, another important parameter that influences the permeation, right? So how it influences? If the surface area is larger and if the vascularity is more, greater vascularity, then the absorption of the drug molecule into the systemic circulation, that is the blood, will be large. On the other hand, if the surface area is less and if the vascularity is low, then the rate of absorption of the drug molecule in that region will be less. So this is how the surface area and vascularity, they can contribute for the permeation. So one example we can take here is that if you take stomach and intestine, so the surface area of stomach will be lesser comparing to intestine. So you know the structure of intestine, right? So which is having a larger surface area. So obviously we can expect more and more amount of drug molecule will be absorbed on intestine comparing to the stomach based on its surface area. Again, surface area is not only factor that is going to that is going to determine the rate of absorption. So we have to consider the pH and pK also. I hope that you remember what we have discussed in the first unit, right? So anyway, in general, if the surface area is larger and the vascularity is more greater, then that can influence enhance the rate of absorption of the drug molecule. Another word that can influence the permeation of the drug molecule. Is that understandable? So for any question, I can answer.
Sir, what is vascularity?
Vascularity is that some tissues will be highly vascular in nature, which means that they will be highly flexible in nature. Okay, so some will be having lesser vascularity, right? Some tissues, some organ will have very high vascularity, and some organs will have less vascularity. Later, I'll give example, right? So while solving the problem for this,
Okay, sir.
yeah. Any other doubt? Okay, assume that no doubt. I go to the next slide. So this topic already we covered in detail, right? So the ionization of the drug molecule. So I already told that most of the drug molecules will be either a weak acid or weak base. We also discussed in detail about what are the functional group which are acid, which are bases. Specifically, which functional groups are weak acids and weak bases. In terms of functional group, we studied this in detail, if you remember, right? So if you take any weak acid or weak base and if you put that in water or any aqueous solvent, so they can either present in its unionized or non-ionized form or ionized form. So this depends on the pH and pK of the functional group. Okay, so this pKa, right? So I did not define what is meant by pKa while discussing the problem, right? So it's the correct time to define what is the pKa. So if we say pKa, just for example, if we say pKa of a functional group is 4.2, again I repeat, a pKa of the functional group is 4.2. So can you tell what is the pKa of carboxylic acid? We solve some problem, no? Can you refer and tell what's the pKa of carboxylic acid functional group? Both acidic and basic functional groups will be having specific pKa. So we solve for aspirin, if I remember. Aspirin will have or carboxylic acid functional group. What's a pKa that we have used for solving the problem? Somebody can
3.5.
respond. Sorry?
3.5. 3.5, sir.
Okay, 3.5. If you say 3.5 is the pK of that functional group, what exactly it mean that at the pH of 3.5? Again, I repeat, you are saying that the pK of the functional group is 3.5. So what is that? What it indicates? So if you keep that functional group in the pH of 3.5, the environment pH of 3.5, then 50% of that functional group will be present in its ionized form, and 50% of that functional group will be present in its non-ionized or unionized form. Does that make sense?
So can you repeat it, sir?
Okay, so let me define, right? So I think without taking the numerical value, if I define, you can easily understand. pKa value is the pH at which the functional group will be present 50% in its unionized form and 50% in its ionized form. So if we say the pK of carboxylic acid is 3.5, just for example. Okay, so if you keep that functional group at the pH of 3.5, 50% of that functional group will be present in its ionized form, and 50% will be present in its unionized or non-ionized form. Is that understandable?
Yes, sir.
Yes, right. So that is what is called as the pKa value. Uh, Again, why this ionization is important, why we studied in detail about the Henderson-Hasselbalch equation. So only the non-ionized uncharged form of the drug molecule can cross the biomembrane. It's a lipid bilayer. So ionized form is better renally excreted because it is more water soluble. Or this point understandable? Very fundamental concepts in medicinal chemistry for understanding the pharmacokinetics. Just 3 points I have discussed. Any doubt in this slide? Any clarification is needed?
So when pK is 3.5 for aspirin, why do we refer it as carboxylic acid, sir?
No, I don't understand the question. Can you say your question again?
So when you ask pK for aspirin,
No, no. I'm PK is not for the entire drug molecule. In aspirin, we have only one acidic functional group. Okay, the PK is corresponding for that functional group.
yes, sir.
Makes sense.
Yes, sir.
If the drug molecule has somewhere if the drug molecule has some basic functional group along with acidic functional group, the pK of that basic functional group will be different.
Yes, sir.
You would understand.
Yes, sir.
So the pK is for the acidic or basic functional group, not for the entire molecule. Understandable?
Yes, sir.
I just told aspirin because it is having only one important weak acidic functional group, that is the carboxylic acid.
Yes, sir.
Yeah,
Understood, sir.
yeah. Any other doubt?
Sir,
Yeah.
can you repeat what's pKa again?
Okay. Okay. Let me. Okay, you take any functional group. Okay, if you take aspirin, so aspirin is having only one acidic functional group. Okay, so that acidic functional group will be having some pKa. Somebody said that the pKa of that functional group is 3.5. 3.5 or 3.4 doesn't matter, right? Just for understanding, 3.5. So if you put aspirin, okay, if you keep aspirin in the pH of 3.5, 50% of the aspirin will be in its acidic form and 50% will be in its conjugate base form. So in other words, 50% will be in its ionized form and 50% will be in its non-ionized or unionized form. So we can define pKa as the pH at which 50 percentage of the molecule will be in its ionized form and 50 percentage will be in its non-ionized or unionized form.
Okay, sir.
Is that understandable?
Yes, sir.
Yes, right? Okay. So any other doubt? Okay, no response. I assume that everyone understands so far. Moving further, in nutshell, ionization concept is very simple. Uh, so ionized drug molecule will be more water soluble, and non-ionized molecular form of the drug molecule will be lipid soluble. That's the only thing you have to remember, right? So it may be confusing, right? So you have to remember this. Only thing is that the ionized form of the drug molecule, the charged form of the drug molecule, will contribute for water solubility, and the non-ionized or unionized or molecular form of the drug molecule will soluble for lipid solubility. Different terms that refer to the same thing: non-ionized, unionized, molecular form, all are same. Ionized, charged, they are same. They contribute for water and lipid solubility. Is that understandable?
Yes, sir.
Okay, assume that that is understandable, right? So moving on further. So this is very simple. The ionization, we will see that later, right? So while discussing the excretion in detail, we will see the renal excretion and all. The point to remember, right? So is that only the free unbound drug molecule can be filtered. I think better I skip this as of now, right? Because we are going to see this detail in the excretion part, right? So I don't want to confuse you. This is very important, right? So we are slowly entering into understanding, uh, what the body does for the drug molecule, right? So once if you take the drug molecule, right? So the drug molecule should be transported, right? So I can give an example for you. So once if you take the oral drug molecule, what is the site of administration?
Mouth, sir.
Mouth is the site of administration. So once if you swallow, right? So once if you take the drug molecule along with water, where the drug molecule will reach?
Stomach, sir.
Stomach, right? So
Stomach,
immediately the
sir.
molecule will move from mouth to stomach. Translocation of the molecule is taking place. Movement of the drug molecule is taking place. So the molecule that is present in the stomach, can we consider that as the molecule present within the body or outside the body?
Within the body, sir.
Any other answer?
Both, sir. Because the intestine only will get absorbed. The rest
Not
will
both,
get
right? So
excreted.
in terms of pharmacology, the molecule is still assumed as it is present outside the body. Again, I repeat, no confusion, right? So once if you take any oral drug molecule, the site of administration of oral drug molecule is the mouth. So we swallow the drug molecule along with some water or some fluid, right? So directly the drug molecule reaches the stomach. So if the drug molecule is present in the stomach, still we consider the molecule is present outside the body, not inside the body. So you may have a question: when we can say the drug molecule is inside the body? So once if the drug molecule entered into the circulation, blood, then we can say that the molecule is present inside the body. Is this concept understandable?
Yes, sir.
Anybody having any doubt in this? Should I repeat again?
Yes, sir. Please repeat it once.
Okay, so this is specifically for the oral drug molecule. We have different types of drug administration. So which means that we have different routes of drug administration. We'll see that later. So some drug molecules are taken orally, some drug molecules are given as injection, intravenous injection. Some drug molecules intramuscular injection. Some drug molecules are used as nasal spray. So these are different mode of administration. If you consider the oral drug, so we keep the drug molecule on mouth because mouth is the site of administration. So we swallow the drug molecule with some water, normally. So the drug molecule directly reaches the stomach through the gastrointestinal tract, GIT. Next class I'll discuss. We'll see the detail, right? So what is that? So directly it reaches the stomach. The drug molecule which is present in the stomach is considered as it is present outside the body, not inside the body. Again, I repeat, the molecule which is present in the stomach is considered as present outside, not inside. So when we consider the molecule to be present inside the body, so only if the molecule present in the stomach enter into the circulation, entering into the blood. Once if the drug successfully entered into the blood, then we can say that the molecule is now present within the body. Is that understandable?
Yes, sir.
Okay, so that's very important, right? Actually, that's a very important question in the core company, right? So pharma company interviews, right? So many students, they assume that once if the molecule present in the stomach, it is inside the body, but in terms of pharmacology, that is not inside the body, right? So how the drug molecule is going to be transported, very important, right? So there are different methods or route. This, I think you might studied this in your either biochemistry or cell and molecular biology, or your 12th standard biology, if you studied biology, right? There are different routes. The three major routes which are used by the drug molecule to transport within the cell or within the body. There, the first important mechanism is passive diffusion. So as I gave example as a room freshener, if you spray the room freshener in one corner, right? So the molecules which are present in the room freshener will start moving. So what's the direction from the higher concentration to the lower concentration? It's a concentration gradient, right? So that same thing that may happen. Passive diffusion is most of the drug molecules will prefer to have this passive diffusion because this is down the gradient, not against the gradient. It's down the gradient from higher concentration to the lower concentration. So what type of molecules can use this type of transport, passive diffusion? So the molecules which are having good lipid solubility, right, they can follow this passive diffusion. So is energy is required for this type of diffusion? No. That is why most of the drug molecules prefer this. So any carrier is needed? No carrier is needed. So is it saturable? No. The movement is not saturable. It will move constantly from the higher concentration to the lower concentration, right? So the second route of transportation is that facilitated diffusion. So this is also down the gradient, very similar to the passive diffusion. Do we need energy for this? No. Any carrier is needed? Yes. For this type of transportation, we need carrier. So some molecule should help this drug molecule to get enter into the cell. Is it saturable? Yes. This type of transport is saturable. Third is very rare. Very few drug molecules will opt for this active transport. The reason for this is against nature, not down the gradient. It's against the gradient. So the movement will be from the lower concentration to the higher concentration. Right? It's against the gradient. And do energy is required for this? Yes, energy is required. So some ATP or some other way the energy should be given for this process. A carrier is needed. Yes, carrier is needed. Is it saturable? Yes, saturable. So more details we will see later, right? So just I am giving some overview, right? So these are the three different mode of transport of the drug molecule, most preferred mode of transport of the drug molecule. So coming to the important topic, right? So as I said that we'll be studying four important parameters in very detailed way in this unit: absorption, distribution, metabolism, excretion. The first important parameter is the absorption. So absorption is something very much related to the permeation, right? So what is meant by absorption? The process of entry of drug into the systemic circulation from the site of administration. I have put very simple definition for understanding. Absorption is the process of entry of drug molecule into systemic circulation from the site of administration. If we take uh, aspirin as example, it is a oral drug molecule and the site of administration is the mouth. Just now we discussed. So what is absorption for absorption of aspirin? So the molecule enter into the stomach. From stomach, it enter into the circulation, systemic circulation, that is the blood. That process is what is called as the absorption. Absorption is the process of entry of the drug molecule into the systemic circulation from the site of administration. Systemic circulation is the blood. So what are the various factors, parameters that decide the absorption, right? So whatever factors we said as the factors which influences the permeation, all those factors can play role for the absorption. So what are the factors we discussed? Solubility, concentration gradient, surface area, and vascularity. All those things you can write for this. All those parameters, factors exactly influences the absorption also. I think again no need to discuss the same point. Whatever I have discussed for permeation is applicable for the factor that influences the absorption. Very important point, right? So very important competitive exam question, right? So the intravascular administration, IV, intravenous, right? So intravascular. So in case of intravascular administration, we inject the molecule directly to the blood. Okay, you had to remember intravenous injection, IV injection. So how doctor place the drug molecule through IV? So they directly place the drug molecule in the blood. Okay, so for that there is no concept called as absorption. If you take oral drug molecule, we place the drug molecule on mouth, it reaches the stomach, then from the stomach the molecule should be absorbed into the systemic circulation. But for intravenous injection or intravascular administration, we directly keep the drug molecule on blood. So there is no concept called as absorption. Is that point understandable?
Yes.
Everyone?
Yes, sir.
Yeah. So now I am going to introduce another important term, what is called as bioavailability. All these are very fundamental things, right? So what is bioavailability? Bioavailability is the amount of drug molecule that actually reaches the circulation. The amount of drug molecule that actually reaches the circulation. Can I repeat? The amount of drug molecule that actually reaches the circulation. I'll give an example for this, right? So far our understanding is that there is no absorption for intravascular administration. So the concept of absorption is only for the extravascular administration like oral drug molecules. First point. Second point is what is the bioavailability? So bioavailability is the amount of drug molecule that reaches the circulation. Again, I repeat, the amount of drug molecule that reaches the circulation. That is what is called as bioavailability. So if you take 100 mg of drug molecule and if you administer that 100 mg of drug molecule through intravenous injection, the entire 100 mg of the drug molecule is placed on blood. Again, I repeat, if you take 100 mg of some drug molecule and if you administer that 100 mg of drug molecule as intravenous injection, what we normally do is that we directly place the 100 mg of the drug molecule on the blood. So what is the bioavailability here? The bioavailability is 100%. Am I right?
Yes, sir.
Yes, right? Okay. So you take another example as oral drug molecule, say aspirin, right? So you're taking 100 mg of aspirin, just for example. So 50 mg of the aspirin is present in its ionized form and 50 mg is present in its unionized form. So which form can enter into the circulation? Which
Unionized form.
unionized. So how much percentage is present in unionized?
50%, sir.
So in terms of mg, milligram, how much concentration? You are taking 100 mg.
50.
50 mg.
50 mg will enter into the circulation. Do you agree?
Yes, sir.
In this case, can we say the bioavailability will be only 50 percentage?
Yes, sir.
Is that understandable or not? Very important concept.
Sir, can you repeat it once, sir?
See, the first point is that, again, I repeat, very important, right? For intravascular administration, intravascular means that administering the drug molecule directly to the blood. For intravascular administration, there is no concept called as absorption, because as per the definition, absorption is the process by which the molecule enter into the circulation from the site of administration. For intravascular administration, the site of administration itself is blood. So there is no concept called as absorption. Absorption is the concept which is related only for the extravascular administration. First point. Second point is, if it is a intravascular administration, the bioavailability will be 100%. The definition of bioavailability is that the amount of drug molecule, the concentration of the drug molecule that reaches the circulation. Since intravascular administration, we directly keep the drug molecule on circulation, the bioavailability will be 100%, right? But if you take extravascular administration, the bioavailability will not be 100% always. What's the reason? If you take aspirin, which is a oral drug, example for extravascular administration, just for example, if you take aspirin, say 50% of aspirin is present in stomach in its ionized form and 50% in its unionized form. So already we discussed that the molecule that is present in its unionized form or the molecule form will contribute for the absorption. So they can enter into the circulation. So how much concentration of the drug molecule now enter into the circulation? Only 50%. So you are taking 100 mg of the drug molecule, aspirin. 50 mg is present in its ionized form. 50 mg is present in its unionized form. The 50 mg present in its unionized form enter into the circulation. So if we make a calculation, what's the percentage of bioavailability? The percentage of bioavailability is 50%. Because we have taken 100% of the drug molecule, only 50% enter into the circulation. So the bioavailability is 50%. Is that understandable?
Yes, sir.
So any questions so far I can answer? Yeah.
I have a doubt, sir.
Yes.
So this bioavailability, does it depend on the presence or absence of absorption, or the drug forms, like molecular, ionized or unionized? On which does it depend, sir?
No, I have taken the ionized, unionized form just as example. Okay. There may be many factors that can decide the absorption and bioavailability.
Yes, sir.
So anyway, the point you have to remember is that for any extravascular administration, the bioavailability will be always less than 100, not 100. But for intravascular administration, the bioavailability will be always 100%. Don't get confused with absorption and bioavailability. Absorption is different, bioavailability is different.
Yes,
Understandable?
yes, sir.
Yeah, okay. So with extravascular administration, less than 100% of dose may reach the systemic circulation because of variation in the bioavailability. See, again, so far any question I can answer, I can give some examples. Anybody
Sir,
having any? Yeah.
does absorption depend on bioavailability or bioavailability depends on absorption, sir?
So here the bioavailability depends on absorption.
Okay, sir. So bioavailability shouldn't be greater than absorption, sir.
Absorption is different, bioavailability is different. Don't get confused. There are
Okay,
different
sir.
parameters that influences the absorption. There are different parameters which influences the bioavailability. We are in the very beginning stage. Don't get confused. Don't think too much, right? So whatever I'm saying, just listen to me. I'll make you slowly understand.
Okay, sir.
Okay. So as of now, you are able to understand the terms, right? What is bioavailability? What is absorption? What is site of administration?
Sir,
Yeah.
I have a doubt, sir.
Yes. Yes.
So, what influences whether a drug molecule is taken in the form of either extravascular or intravascular, sir?
That again depends. Okay. Say for example, uh, okay. So say for example, near intestine. Just layman example I'm giving, right? So everyone should understand. I'm not giving very scientific example, right? So we will see later. Say example, you have a drug molecule A, some drug molecule A. So unfortunately, if you administer the drug molecule A as a oral drug, so the molecule is not highly stable in the gastric juice, which is the content of stomach, which means that the molecule break down. Okay, if that is the case, what will be the bioavailability?
0%, sir.
Not zero, it will be very less. Am I right?
Yes, sir.
Okay, it will be very less, right? We cannot say whether all the molecules will be degraded. It will be very less, right? So in that case, you know that the molecule is having the ability to bind to the target to produce the desired biological response. But unfortunately, if you give it as a oral drug molecule, the molecule is not highly stable enough in the gastric environment of the stomach. So what is the problem now?
It cannot
The
be taken orally, sir.
Sorry,
It cannot be taken orally.
it cannot be taken orally. So can we say the route of administration is not correct for this drug?
Yes, sir.
So if we make that molecule as a liquid, and if we inject that molecule directly to the blood, do you think that blood will be having the same gastric environment like the stomach?
No, sir.
We can bypass that problem, right?
Yes, sir.
That is how the company will decide which mode of administration is better. You will understand.
Yes, sir.
That is a question, right?
Yes, sir.
I'll give one more example, right? So you have some block on your nose, or some infection, nasal infection. So the company will go for inhalers, or it should come up with oral drug molecule or intravenous injection. Which one do you think is best?
Maybe inhaler, sir.
Inhaler, right? So different purpose, right? So I hope you got the point, right?
Yes, sir.
There is no one straight reason or point to say that whether we should go for oral, right? So maybe different things they may consider to say that which mode of administration is the best. But for normal systemic thing, the company will prefer to go for oral drug molecule, because that is profitable, and that is easy for administering, right? So the person can easily take the drug molecule. There is no issue of going for a nasal spray or any other. But if it is a kind of injection, intramuscular, subcutaneous, or intravenous injection, you may need some support from others. But there are drug molecules which cannot be made as oral drug molecule, can only go for intravenous injection or some other type of injections. There are different reasons there. We'll see that later. Okay.
Okay, sir.
Yeah. Any other doubt so far? Anybody having any doubt? Okay, I assume that no doubt, and I move to the next slide. Very important slide. What is bioavailability, right? All these are very fundamental things in terms of drug design or medicinal chemistry, right? So whenever you talk to any expert in the drug design field, right? So as I said that we are running some company, we are we have a startup, right? So we are in the process of discovering many drug molecules related to microorganism, and bacterial or fungal, right? So whenever you talk to any expert in the field, right? So say if you talk to any person from Pfizer or Novartis, right? First question what they will ask, okay, you have some new molecule, what is the bioavailability of the molecule? Okay, this is a very fundamental thing but most important thing, right? So let me clearly tell what is the bioavailability, right? So this is a graph drawn between time on x-axis and the drug effect on y-axis, or cp here is the plasma concentration. Again I repeat, so the simple graph that is drawn between time on x-axis and the plasma concentration cp, drug effect on the y-axis. Cp is the plasma concentration of the drug molecule in the plasma, right? So what is this plasma? Plasma is a component of the blood. So normally if you go for any blood test, they will collect the blood from you. So they will insert the needle intravenously, and they will pull some quantity of the blood from your body, right? So they may want to measure the presence or absence of something, right? But they won't take the whole blood. They will centrifuge the blood. Okay, once if you start centrifuging the blood, so that will separate two layer. Okay, one layer is some yellowish fluid, and that fluid is what is called as the plasma. Plasma is a liquid component, watery component of the blood. That is the plasma, that is on the y-axis, right? uh, so here probably this graph, the route of administration, once if you have the time versus plasma concentration, we can easily tell what is the route of administration. So here this is the time 0, initial time, and this is after some time. Okay, so increasing time on the x-axis, so increasing the plasma concentration on the y-axis. So this graph should be probably for oral administration, right? So just by looking at the time versus cp, we can say whether it is a oral administration, extravascular or intravascular administration. So this is definitely example for extravascular administration, that is the oral administration. So how do we say it is a oral administration? Because at time 0 the concentration, plasma concentration is 0. So which means that once if you take any oral drug molecule for few seconds or few minutes, the initial concentration will be 0. Why the initial concentration is 0? Because no drug molecule till enter into the circulation. So the plasma concentration will start increasing when the drug concentration increases on the blood. So initially the concentration of the drug molecule is 0. When time goes on, right? So when time goes on, say 3 minutes, 5 minutes, 10 minutes, 15 minutes, 30 minutes, the concentration increases. The plasma concentration increases. So the plasma concentration reaches a peak, the maximum. Okay, then there is no more increase in the plasma concentration. The plasma concentration slowly started to decrease. There is a decline. The plasma concentration goes down, 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 reaches closer to 0. This pattern is example for oral drug molecule. Is that understandable?
Can you repeat it again, sir?
No, I did not repeat the entire graph. So far what I said is understandable. Okay, I repeat again. So this is a graph plotted between time and the plasma concentration of the drug molecule in plasma, which is nothing but something like concentration of the drug molecule in blood. As a beginner, you can understand this as the concentration of the drug molecule in blood, time versus concentration graph. So this graph is specifically made for a drug molecule that is administered orally. So how do I say that this is corresponding for oral drug? Because time 0, the plasma concentration is also 0. Again, I repeat, time 0, the plasma concentration is also 0. When time goes on, the plasma concentration increases. Okay, so after reaching certain point, after time x, there is no increase in the plasma concentration. The plasma concentration started to decrease or decline. Okay, reaches down, 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 closer to 0, and this is the pattern for a oral drug molecule. Is that understandable?
Yes,
Again, I
sir.
did not explain the entire graph. We will explore slowly, right? So okay, so this dotted line here, I hope my cursor is the laser pointer is visible, right? So this dotted line is the uh, minimum effective concentration. Again, I repeat, this is the minimum effective concentration. So which means that your body has some pain. You're taking a painkiller. Okay, so say for example, you have taken the painkiller, so the concentration is up to this. Okay, you cannot feel that the pain is relieved. Even though some concentration of the drug molecule is present in the blood, in the plasma, we cannot expect reduction in the pain. Why? Because if you want to feel reduction in the pain, the minimum concentration that is required is this point. This is the minimum concentration that is required to see the desired effect. Is the point understandable? Should I repeat or
Understood, sir.
everyone?
Yes.
Okay.
Yes.
So once if the concentration goes beyond this minimum effective concentration, that is the moment you will start feel that the pain is reduced if it is a painkiller. Okay, that is why you have to take the correct dose. You are taking some drug molecule. If the dose is not sufficient enough to reach this point, what's the value of taking the drug molecule? There is no value. There is no use of taking the drug molecule because the concentration you have taken is not able to reach the minimum effective concentration. So whenever you take any drug molecule, you have to think of that the concentration of the drug molecule should cross the minimum effective concentration. Then only it can produce the desired biological response. Is that point understandable?
Yes, sir.
One more point. Slowly we are exploring the graph, right? So one more point is that this is the maximum concentration we can use. So once if the concentration goes beyond this dotted line, okay, the drug molecule will start producing the adverse effect. It's the maximum effective concentration. It's the minimum effective concentration. It's the maximum effective concentration. Okay, the window that is between the maximum effective concentration and this minimum effective concentration, that is what is called as the therapeutic window. Again, I repeat, the difference between the maximum effective concentration and the minimum effective concentration is called as the therapeutic window. So always we have to maintain the concentration between this maximum effective concentration and the minimum effective concentration. The concentration should not go below the minimum effective concentration, and also it should not go above the maximum effective concentration. Because if it goes below the minimum effective concentration, the drug cannot produce the biological response. If it goes beyond the maximum effective concentration, the drug molecule will start producing some adverse response. Is that point understandable?